guys, my name's Susanne. I work at the chemistry department at the University of Oxford in Carol Robinson's lab.
Hello, my name is Jussi. I'm at the Nuffield Department of Clinical Neurosciences in the Becker lab.
And we are both postdocs in the Kavli Institute. We're hoping to do this as a series of videos showing you all the science that's going on at the Kavli, what sort of exciting research is being done, and also what sort of machines we use, what sort of techniques to really understand our bodies and our world.
Yeah, and we're starting with your work today.
We're starting with my work, yeah. So you are made up of nerves.
Mm-hmm.
I mean, there's lots of things in your body that aren't nerves, but everything you touch and feel and smell and see and hear, all of that is happening with nerves. And any reaction you have to that, I mean, any thought or emotion you have is nerves in your head communicating with each other, which we call the brain. Nervous communication has to be really fast. So if I touch you,
it's immediately in my brain.
You know, it's immediate, so it goes really quickly through the nerve.
Yeah.
But then to connect with your brain, it has to move from one nerve cell to another nerve cell. So the first nerve cell releases some chemicals, we call them neurotransmitters, and the second cell has to detect them. So the second cell has a series of proteins all around the end that all detect different chemical signals. And so depending on what chemical signal and how much, you get a different reaction in your nerve, and you might smell oranges, you might hear a plane, or you might want to cry.
Yeah.
So my work is focused on some of these receptor proteins that is in the kind of second nerve cell, and how they work and how they can go wrong and how that can cause different issues, basically.
Yeah. And without having these synapses or these junctions between two neurons, we couldn't have any kind of control over what's happening in the nervous system.
No control, you wouldn't be able to move any of your muscles, you wouldn't be able to feel anything.
Yeah, because if you started an electric impulse, that would just run through the whole system without these breakpoints.
Yeah.
How do you study these proteins, or how do you study these synapses?
I weigh them.
But you can't obviously use a scale, so how?
No, they're very, very, very small, so you need a really, really tiny scale.
Yeah.
No, we use something which is much bigger than a scale. You'll see them in the lab in a minute.
Yeah.
It's called a mass spectrometer.
So what do you get out of weighing these proteins? Or?
That's a good question, because really, I mean, if I want to know the weight of any protein in your body, I can just look it up on the internet.
Yeah.
We know how much proteins weigh. And the point is, if we can weigh them precisely enough, then we can see a lot of what's going on with a protein. So for example, if a protein has been modified and that makes it hyperactive, that'll change its weight.
Yeah.
Or if a protein has a bit broken off and that makes it not respond to the chemicals, then that will change its weight.
Yeah, that's really interesting.
I think so.
Yeah. So before we go in,
Okay.
can you tell me what exactly it is that you're studying?
Okay, so we said that the second nerve has all these different detector proteins. I work on one class of these detector proteins, so they all react to serotonin, which is, you might have heard, kind of called the happiness molecule. It's involved in a lot of functions in your brain. Interestingly, 90% of your body's serotonin is in your gut, and another like 5% or so is in your blood. And there's only a tiny bit of it in your brain, but the tiny bit of it in your brain is really important for your brain.
Yeah.
So studying serotonin and the proteins, so serotonin gets released from the first nerve, it's one of these chemicals, and then it gets detected by the second nerve. And I'm looking at the proteins that detect it. And actually one of the proteins that detects serotonin, when it detects the serotonin, it opens what's called an ion channel to let ions into the cell. And that is my favorite protein. So this is our mass spectrometry lab where we keep all of our mass spectrometers. We've got approximately 1, 2, 3, 4, 5, 6, 7, 8 or so.
Oh, that's
maybe some I don't know about.
Yeah.
And they all have slightly different arrangements and they all do slightly different things. And so altogether we can do quite a lot of mass spectrometry.
Yeah.
So this is the one we'll be looking at a bit today.
Okay.
It's actually an older model that isn't currently in use in the lab, but that's why we can kind of take it apart and get inside a little bit.
Yeah.
So whatever we want to weigh in the mass spectrometer, we put into one of these needles, which is a glass needle, and then it's been coated with gold on the outside.
Yeah.
So for example, if I have my protein sample here,
It's a very tiny tip.
yeah, so we need to get all the way down to the tip of the glass capillary, otherwise the sample will get stuck in the middle. So get used to doing this over time, but you never get it on your first try.
Yeah. Oh, it's almost there. Yeah.
There we go, and then we can load the sample into the needle, and then we're ready to take it to the machine. So when we have our sample in the needle, we can attach it into the front of the instrument, and then when we apply a voltage to the needle, my liquid sample becomes a gas
Oh.
and gets sprayed into the instrument.
Okay.
And then it travels through, so we can see actually the path of my gas through the instrument in a
That's
minute.
cool.
You want to help me with the panel?
Oh yeah, yes, please.
So the liquid sample comes from the needle, it enters the machine here as a gas, and then it goes through a bunch of different chambers that we'll talk about in a minute, until it gets to here. This is where the proteins are actually weighed. This is a big tube that goes all the way down into the machine and up again. And actually the gas is sent all the way down and all the way back up again to weigh your proteins.
Oh, so what is this?
So, inside the machine, all we could basically see was a bunch of boxes in a row.
Yeah.
This is one of the boxes that's inside the machine.
Okay.
And inside the box,
Ooh, that's
yeah, this is where the magic happens.
cool. So your sample comes in to this bit?
So a mass spectrometer is mostly kind of two parts.
Mm-hmm.
One part is the bit right at the end that actually weighs the ions. That's the big tube in there,
Yeah.
or it can be other bits in other mass spectrometers.
Yeah.
But on the way there, you have a first bit, which is basically to focus and clean up your gas sample.
Mm-hmm.
So when you spray it in, it's just a big mess of gas. And
So
you
it's
need to,
just all floating around in the machine.
yeah, exactly. And you need to get it kind of going in the correct way and focused into a nice tight beam of ions and going where you want it to. And you need to clean up, for example, some of the bits that you don't want, they get taken out in the process.
And how do you accelerate the sample?
So what you need to be able to do mass spectrometry is you need your sample to have a charge. So it needs to have either a positive charge or a negative charge. And once it has that, you can use magnetic fields and radio fields to drive the ions because the charge reacts to a charge. So quite often in a mass spectrometer, for example, you'll have a bent bit where you're bending the ions around a curve. And anything that doesn't have a charge will just keep going straight forward. So that's how you get rid of anything that doesn't have a charge.
Yeah.
And so you have all these different kind of boxes in a row that are to get rid of unwanted bits and to focus on your sample so that when it gets to the weighing machine, it can be weighed more straightforwardly.
Okay.
And the reason you have to have several boxes, chambers would be the technical term. The reason you have to have several chambers in a row is because you actually need to get your sample down to quite a high vacuum.
Mm-hmm.
So for example, in some of our machines, the vacuum in the weighing chamber where the proteins get weighed is almost as low as on the moon.
Okay. That's cool.
Yeah, and you can't just go from like room temperature down to moon atmosphere in one step.
Yeah.
So you have a couple of these in a row almost. And every time you enter a new one, the vacuum gets
Lower.
a bit more vacuumy.
Okay.
And then your gas will tend to expand. So then you need to focus it again,
Okay.
and then it tends to expand. So the way we focus these ions,
Oh, that looks.
this is how you focus an ion beam.
Okay.
So this is called a quadrupole because it's got 4 poles. And basically, all of this is to make the right magnetic and radio field in the middle so that the ions will be sent through the middle and they'll be kept focused. And any ions that you want, they'll be sent straight through the middle of the pole. And any ions that you don't, they end up wiggling enough that they get shot off to the sides.
Okay.
And either they hit the poles or they come out, they get gotten rid of.
And that's inside a vacuum.
That's inside increasingly more vacuum,
Okay,
basically.
yeah.
So you'll have one of these, and then you'll have a lens of some sort, which is basically just a hole.
Mhm.
But again, because you've got magnetic fields being created, when it goes through the hole, all the ions are affected.
Okay.
So you'll have a quad, and then a lens, and then maybe a different bit with some different focusing, and then another lens. And then by the time you get down to your weighing machine, your ions have all been beautifully,
Filtered out.
yeah,
Yeah.
basically.
So do all the chambers have a similar kind of thing? Like this? Because those look similar.
Yeah, so this is a quadrupole in here, and this is a quadrupole here, which is a really, that's like a heart of a mass spectrometer, but there's a lot of other things you can use as well. So this, for example, is very similar to a quadrupole, but it's got 6 poles,
Oh, yeah.
so it's called a hexapole. And it's basically a similar way of doing the same thing. We also have some other bits that can be used to kind of clean up your sample or change your sample in different ways. So this, for example,
Oh, that looks cool.
is a ion mobility cell.
Yeah.
And again, the ions just go through the hole in the middle. So the neat thing about a mass spectrometer is your ions are just going through, it's like they're just going through space. But we have all these electronic bits to create the magnetic fields that make them go where we want to or make them behave like we want to.
Yeah. It's like a train on a train track.
Yeah.
It's going in a certain direction that you tell it to go to.
Yeah, and you might have complicated train controls,
Yeah.
but the train can only go one way.
Okay, that's cool.
So all of that, as we kind of said, is only to clean up your sample. But how do we actually weigh the ions?
Yeah.
Okay, so when we want to go into the mass spectrometer, we have, as we saw earlier, your sample is loaded into our little glass needle, and then the ions are sprayed out of that needle into a gas, and then they enter the machine. So inside the mass spectrometer, we can have lots of different elements in lots of different orders. So for example, we might have a quadrupole, which filters the ions and gives them the right energy to get through the rest of the machine. And then you might have a lens that focuses the ions, as we said, to get into the next chamber where the vacuum is lower. And there we might, for example, have a hexapole, which again filters the ions and gets them moving in the right direction. And you have a series of these lenses and quads and hexapoles and so on. And then you get to the really interesting bit, which is where the ions are weighed. So this is the big tube that we saw inside the machine. And what happens here is that the ions enter at the top. So they go all the way down and then all the way up to the top again. And then they hit a target, which is the detector, which is where we actually detect the ions.
So how are the ions accelerated?
You can think of it like a sailboat race. So all the sailboats have a certain size, and the bigger the sailboat is, the slower it goes, right? I mean, like, the more you need to put energy in it to get it to go. But the bigger the sail, the faster it goes. So all the sailboats, all the ions, they get the same acceleration from the machine. But if you've got a really big sail, you'll go faster. And if you're a really big ion, you'll go slower. So
the size of the ion and the charge on the ion.
So the charge is like the sail,
Yeah.
because it's using the charge to accelerate.
Yeah.
So the bigger the charge, the faster the ion will go.
Okay.
But the bigger the ion, the slower the ion will go.
Yeah.
So the order, like which order they get to the target in, is dependent both on the size and on the charge. So when we say that we're weighing proteins, the thing that we're interested in, as we spoke about earlier, is finding out the size of the ion, the weight, because then we can tell all sorts of interesting biological things. The thing we actually measure is the weight divided by the charge, which makes everything a little bit more complicated. But luckily we have lots of ways to figure out from this value what the weight of the protein is, and then we can do science on that.
Yeah.
And that's it. So thank you so much for coming to my lab. I actually only just joined this lab in January, and I'd never even touched a mass spectrometer before then. So this has been a big learning curve for me. And just a little while ago, I got my first mass spectrum.
Yay!
So I'm really excited to be learning new skills and trying new things at the Kavli.
And that's what we do here at the Kavli. I'm actually a medical doctor, and I'm switching to a new field in neuroscience. Now, in our next video, we'll be working with brain organoids that we're making out of stem cells.
That's really cool. I can't wait to see it. Okay, so I'm Susanne.
And I'm Jussi.
And just at the end, we want to say a massive thank you to everyone who's helped out with this video, to our funders who fund our research, and specifically
to you for watching.